Mutations, cause and effect. So I just wanted to make a quick note about transcription. I want to point out that RNA and DNA are very similar, but RNA does not have thymine. So instead of thymine, it has uracil, and therefore adenine binds to uracil. So in transcribing DNA to RNA, here we have a DNA code, CCATT. We transcribe it to GGUAA. During translation, the mRNA leaves the nucleus and protein is formed in the cytoplasm as the ribosome reads the 3 RNA-based codon and finds the tRNA with the complementary anticodon that carries a specific amino acid. So when you are trying to decode the mRNA and figure out what amino acids link together to make the protein, you can use something like this, like a codon map or a codon chart. Here you map the codon to the appropriate amino acid. So let's start out with a codon. We're going to be looking at GCC. You find the first nucleotide, the G, in the center here, and then the second one in the second circle, C, and then follow the third one into the third circle, C, and you find here that alanine is the amino acid that that codon is searching for. I want to make a note that the first 2 nucleotides in the codon are strict, and they often indicate a distinct amino acid. And then the final nucleotide has some redundancy, where several different letters result in the same amino acid being used. For example, here, GCC and GCG, GCA, GCC, and GCU all code for alanine. Now, mutations can change the amino acid used in the protein, which can result in the protein folding slightly differently and potentially causing a problem in the cell. There are several different types of mutations, including substitutions, insertions, and deletions. A substitution is when a single nucleotide changes. This is often the result of miscopied DNA or RNA. Here you see that A turns into a G. Insertions are when extra nucleotides are pasted into DNA or RNA. Here the TGG gets inserted. And deletions are when nucleotides are cut out of DNA or RNA. Here the 2 Gs are removed. Finally, a frame shift is the result of an insertion or deletion, and it's when the entire reading frame changes, and it results in a non-functional protein sequence. A good way to think of this is like this sentence here, where you have the 3 letters, like each of them the codon, and you can read them as a sentence that makes sense. The fat cat sat. However, when you delete the T, and then you take the 3 letters and try to read that, it becomes gobbledygook. This sentence does not have any meaning, and this is kind of what happens when there's a frame shift in reading the mRNA. So mutations can result in several different outcomes. Here we look at point mutations. For example, a silent or a synonymous mutation is when the codon changes but the amino acid stays the same. Synonymous indicates that it has the same meaning. In this case, the genotype, or the code itself, changes, but the phenotype, or the observable trait, remains the same. So if we look at this example, first we'll look at the column with the no mutation. We're looking at the DNA level is TTC, the mRNA level is AAG, and the protein level is lysine. It pulls lysine as an amino acid. And this is what the original code should code for. Now, in the silent point mutation, that DNA level changes, and the third codon is a T, which changes the mRNA to AAA, but it still pulls the same amino acid, lysine. There are also non-synonymous mutations, where the meaning changes and the genotype and the phenotype change. So one type is the nonsense mutation, when a mutation results in a premature stop codon and truncates the protein. It's usually rendering it non-functional. So here in this example, the nonsense, the DNA level, the first codon changes to an A, which results in a UAG at the mRNA level, which pulls in a stop and truncates the protein. Also, another type of non-synonymous mutation is a missense mutation. And this is where the amino acid changes. And there's two types here, where the missense can be conservative when the replacement amino acid has similar chemical properties and thus functions in the same way as the original code. In this example, the original code pulls a lysine, and it has this purple square around it, which indicates that it's basic. And the conservative missense mutation pulls an arginine, which is also a basic amino acid. So therefore, the function may actually stay the same because the protein may fold in the same way. However, you can also have a non-conservative missense mutation where the alternate amino acid has different chemical properties and thus will not behave like the original protein. So here in this example, the non-conservative mutation pulls in a G, changes the amino acid, which is a threonine, and that is has a polar entity on the end, which may change the folding and therefore the function of that protein. So I should note that although mutations are often detrimental, not all mutations are bad. They can result in a trait that helps an organism survive. And I wanted to also point out that some mutations can be inherited. For example, mutations that occur in the sperm or egg can be passed on to future generations. Other mutations are accumulated in one's lifetime and may affect that individual, but do not get passed on to mutation. Because different genes code for different things, the mutation rate among genes can vary a lot. Let's take a look at this. So this is a table of substitution rates in mammalian genes per billion years, and it's looking at non-synonymous substitution rates and synonymous substitution rates. So remember that the synonymous mutations have different nucleotides but end up with the same amino acid, and the non-synonymous mutations result in different amino acids. So if we look at this example here, H4, which is a histone, the non-synonymous mutation rate is 0 per billion years, which is obviously very small. And then the synonymous substitution rate is a little bit higher at 3.94 per billion years. So if you look closely at this table, you can see that the rate of non-synonymous substitutions in this column is much lower than the synonymous substitutions here going down. All of these are lower than all of these synonymous substitutions. And why is that? If we think about it, this makes sense because the mutations that cause the proteins to change, these non-synonymous mutations, may cause problems with the function of a cell. These mutations will therefore lower the fitness of those individuals, and the mutations themselves will drop out of the population. And in terms of synonymous substitutions, these are mutations where the amino acid itself doesn't change, and so therefore the protein stays the same, the function stays the same, and any kind of correction mechanisms may not actually find those synonymous mutations. And so they stay in, so you see that there's a little bit more of them. Another interesting note about this table is that you can see that the histone and ribosomal proteins have very low non-synonymous substitution rates, and the interferons have a slightly higher non-synonymous substitution rate. So what's going on here? This is likely due to the fact that histones and ribosomal proteins are involved with translating RNA to DNA. So if they have a change in the protein sequence, it will likely be detrimental to the cell. However, interferons are involved in the response to viral or bacterial pathogens. In this case, some changes in the protein sequence may actually be favored by natural selection because it may extend protection against similar strains of pathogens that come in, these interferons can actually find them. So learning about the rate and type of mutations can tell us something about selection at the level of the gene. So with histones and ribosomal proteins, that selection is really strong, so they have to stay the same. There can't be a lot of changes. And then interferons, the selection is a little bit looser. There's a little bit less selection, and it allows to have more non-synonymous mutations. I want to make a final note about terminology that will come up a lot in this class and that I briefly mentioned in this video already. So genotype and phenotype. A genotype is the actual genetic code, and phenotype is the observable trait. So here, the genotype, the code, and the phenotype is the trait. So the trait would be something like maybe size or maybe coat color here for this rodent. And then identical twins share the same genotype, but environmental factors may cause mutations or differences in gene expression that result in a slightly different phenotype. And you can kind of see here, this just gets kind of a superficial example, but that even identical twins don't look exactly the same.